Hello, everyone, and welcome to the Ethel Browne Harvey Postdoctoral Seminar Series. My name is Talia Hatkevich, and I am a postdoc at Duke University, and I will be moderating today with Gerrald, a postdoc at University of California, Santa Cruz. We are excited to highlight the work of our outstanding postdoctoral members. Today, Rui Yan from Harvard Medical School and Taylor Medwig-Kinney from University of North Carolina at Chapel Hill will share their research. Each speaker will give a 20-minute talk followed by 10 minutes of Q&A. Please remember to enter your questions in the Zoom Q&A box. As the first moderator today, I am honored to introduce our first speaker, Rui Yan. Rui received his PhD at UC Berkeley, where he developed super-resolution microscopy techniques to study cytoskeletal biology and protein trafficking. From there, he joined Cliff Tabin's lab in the genetics department at Harvard Medical School. As a postdoc, he continued developing his microscopy skill set, applying it to novel ex vivo culture systems for visualizing morphogenesis, which is what we will hear more about today. Throughout his young career, he has demonstrated uber productivity as showcased by his 20 publications. He was also awarded with the prestigious Helen Hay Whitney Postdoctoral Fellowship. Lastly, I asked Rui what he finds most rewarding about scientific research, to which he responded, understanding how biological systems work. This response really underscores his love for curiosity-driven science. And with that, let's all welcome Rui. Rui, take it away.
Thanks, Talia, for the kind introduction. Let me share my screen. I'd also like to thank the organizers for inviting me here to share my work. I'm really interested in epithelial morphogenesis and how it shapes organ development. Today, I'm going to talk about the biophysical mechanism of foregut tube splitting and how it affects some congenital birth defects. In this video, we are seeing the process where a continuous epithelial tube in the foregut splits into two tubes to give rise to the trachea and the esophagus. This process, called tracheoesophageal separation, or TES in short, is a critical embryonic event whose failure can lead to life-threatening birth defects. TES starts with the dorsoventral patterning of the embryonic foregut, with the dorsal cells in green developing into the future esophagus and the ventral cells in purple developing into the airway. Next, a pair of lung buds will emerge on the ventral side of the foregut to become the lungs, while the trachea and the esophagus are formed concomitantly by splitting the upper foregut tube. When TES goes awry, the embryo can develop a range of congenital malformations affecting 1 in 3,000 newborns. These malformations are lethal and require immediate surgical intervention. As you can imagine, the food taken by the affected infant could end up in their lungs. However, we still know very little about their developmental mechanisms. What we do know are the genes and signaling pathways that pattern the foregut tube. Here is a cross-sectional view of the embryonic foregut right before TES, showing the dorsoventral patterning of the epithelium determined by the morphogen gradients in the surrounding mesenchyme. However, we know very little about how these signaling pathways and morphogens determine the tissue morphogenesis in TES. Beyond its critical role in embryonic development, tracheoesophageal separation also provides a unique system for studying epithelial morphogenesis involving topological transitions. It's different from the well-studied systems like epithelial tube branching or epithelial folding because in TES, it involves an epithelial splitting where one tube becomes two tubes. In topological terms, this process is not homotopic, meaning there's no continuous transformation to make two separate tubes from one single tube. And understanding the biophysical mechanism of epithelial splitting will give us a novel insight into epithelial morphogenesis in general. The conventional way of studying mammalian tissue morphogenesis is to look at fixed tissue sections at different sequential developmental stages. And using this approach, previous studies have identified the key steps in TES, starting with the bilateral narrowing of the foregut epithelium to the formation of the epithelial junction called the septum, and the resolution of the junction to form two separate tubes. However, there are still a few key questions unanswered, including what kind of biophysical forces induces the initial epithelial constriction, and how is that force generated, and how pathological TES happens. To address these questions, we decided to analyze live tissues so that we could access the biophysical force, and we compared two species, the mouse and chick. Both species undergo TES, but they exhibit differences in their foregut morphology. And we reason that by finding the common mechanisms between these two species, we would identify the evolutionarily conserved and essential features of TES. So we started by analyzing the force balance at this epithelium-mesenchyme interface. As TES happens on the timescale of hours, at any moment we can treat the tissue as close to force equilibrium. And that leads to two possible models. In the epithelium-driven model, the epithelium itself is actively constricting, while the mesenchyme must be pulling on it to balance the force. And in the mesenchyme-driven model, the epithelium is pushed by the surrounding mesenchyme and resists that force. To distinguish these two models, we cut a live foregut slice from the chick embryo and removed the surrounding mesenchyme. What we saw was that the epithelium expanded immediately, suggesting it was originally under constrictive force. Overlapping the two images shows that the expansion is more dramatic in the medial region, suggesting the force might be biased or enriched here in the middle. In case that we accidentally stretched or torn this epithelium, we also performed laser ablation to remove mesenchymal tissue locally. Here we ablated a few cells in this magenta area, and after the ablation, we saw an immediate expansion of the epithelium toward the mesenchyme, again supporting the role of the mesenchymal force pushing the epithelium. So this compressive force could, in principle, constrict the epithelium and help in TES, but whether it is really important. So to test it, we need a system where we can monitor the tissue morphogenesis after perturbing the mesenchymal tissue. So we put this freshly cut foregut slice into the matrigel and cultured it. And this simple culture system worked really well in recapitulating TES in both chicks and mice. Here in these videos, we could see all the essential steps of TES from epithelial narrowing to tube splitting. And the bonus point is that we also get unprecedented spatial temporal resolution at the cross-sectional level, which really allow us to decipher the cellular dynamics in it. And with this system, we could finally test whether the mesenchyme is important by culturing the mesenchyme reduced or removed slices. Here, when the mesenchyme is partly reduced or almost completely removed, we saw that TES could not complete, and instead the tube relaxed into a single tube. And this data suggests or demonstrates that the mesenchymal force is indeed essential in TES. To more quantitatively characterize the morphological evolution of the epithelium, we developed an AI-assisted segmentation and quantification pipeline where we use the segment anything model to extract the contour of the epithelium and track its evolution and measuring various morphological traits. The first parameter we took was the width of the epithelium defined by the distance between the two curvature minima. And we aligned different samples by the time of formation of the septum. Before the formation of the septum, the width exhibits a slow decrease, and after the formation of the septum, it decreased more and more rapidly. In the mesenchyme removed sample, this width does not decrease at all. The second parameter we took was the perimeter of the foregut epithelium divided by the square root of its area, which approximates how elongated the shape is. For a circle, this value is about 3.5, and the larger value indicates a more elongated shape. In the control, we could see this ratio increased at an accelerating rate, while in mesenchyme removed tissues, this shape remained at a shape close to a circle. These results further corroborated the critical role of mesenchymal force in initiating TES. And next, our question is, what is the physical basis of the force? So for the sake of time, I will skip how we ruled out a few possibilities and instead focus on a few lines of evidence showing that there is a convergent flow of the mesenchymal cells that is critical to the mesenchymal force. First, we performed particle image velocimetry to infer cell movement at the tissue level. This works by computing the image correlations between adjacent time points. And here, these arrows indicates the directions and the speed of tissue movement. In both chicks and mice, we could observe a highly convergent flow of mesenchymal cells toward the epithelium. And this convergent flow can potentially generate a squeezing force to constrict the epithelium. Moreover, for images that have good nuclear segmentation, we also performed individual cell tracking to see the trajectories in these segmented cells. And then we quantify the moving directions of cells in the blue and red boxes on the left and right sides. We find that the cells on the left in the blue box are moving toward right, and the cells on the right are moving left, again supporting a convergent flow of cells. And if the directional cell migration is critical in this mesenchymal force, we should be able to block it by inhibiting cell migration. Here we use the small molecule inhibitor of the focal adhesion kinase, which is critical to cell migration and is abundant in the foregut mesenchyme. With this inhibitor, TES was stalled. And quantitative analysis showed that TES stopped after an initial narrowing of the epithelium. Although we can't rule out that inhibiting focal adhesion kinase may have other biological effects than disrupting cell migration, we performed a mechanical rescue experiment which shows that an external bilateral mechanical force can rescue the focal adhesion kinase defect. To do this, we made a narrow slit in a collagen gel, which is different than the tissue slice, so that it requires a little squeezing of the tissue to fit it in, thus generating a bilateral force. We could see that compared to the control sample without compression, which cannot separate with the focal adhesion kinase inhibitor, the compressed sample could indeed complete TES. So that suggests the convergent flow is due to the directional mesenchymal flow. And next, we want to understand why the cells are moving in that way and what is the molecular basis of such directional flow of cells. And by reading into the work describing human birth defects as well as mouse mutants with TES defects, we landed on sonic hedgehog. Sonic hedgehogs is a morphogen secreted by the epithelium and sensed by the mesenchyme. And it was shown more than 20 years ago that sonic hedgehog knockout embryo in mouse could not finish separation. Sonic hedgehog signaling is also implicated in human patients where its downstream effector genes GLI3 and FOXF1 were found in human patients with related birth defects. So we first confirmed this critical role of sonic hedgehog signaling in the chick model using a pharmacological drug inhibiting sonic hedgehog signaling, the cyclopamine. And with this model, it didn't perturb the dorsoventral patterning of the foregut dramatically, but the mesenchyme was greatly reduced in both chick and mouse. And live imaging shows that in sonic hedgehog deficient slices, the epithelium is stalled at the narrowing phase, and the mesenchyme lose their convergent migration toward the epithelium. Quantification suggests that the morphological traits of the sonic hedgehog inhibition closely resemble that of the mesenchyme reduced samples. And to further understand which cell type mediates sonic hedgehog dependent convergent flow, we perform single cell RNA sequencing because the mesenchyme itself is also patterned. And here, using single cell RNA sequencing, we identify the major cell types in the mesenchyme here illustrated. And then we plotted the cell composition between the control and cyclopamine-treated samples and identified this yellow cell population, the dorsal subepithelial mesenchyme, being the most dramatically affected cell type in the cyclopamine treatment. Immunostaining further confirmed that this cell type, labeled by NKX 6.1, is completely absent in sonic hedgehog inhibition. And these results suggest that sonic hedgehog is critical to the development of this cell type. And we further showed that this cell type mediates the convergent flow and the mesenchymal force to facilitate TES. Finally, we want to understand whether sonic hedgehog itself can attract mesenchymal cells. To do this, we implanted a bead loaded with sonic hedgehog into the mesenchyme and analyzed cell orientation. This is done by computing the angle formed by the Golgi apparatus, the nucleus, and the center of the implanted bead. Because in persistently moving cells, the Golgi is always at the front of the nucleus. So if this angle is smaller than 90 degrees, that suggests the cell was oriented toward the bead. And when this bead was loaded with saline, the distribution of these angles is not significantly different from an uniform distribution where the cells are randomly oriented. Well, with a sonic hedgehog loaded bead, we see a skewing of the distribution toward angles smaller than 90 degrees, which suggests sonic hedgehog can directly induce mesenchymal cell polarity. We also quantified cell orientation at the endogenous epithelium-mesenchyme interface. And here, the angle is defined by the Golgi nucleus and the interface between epithelium and mesenchyme. Again, if this angle is smaller than 90 degrees, it means the cell is oriented toward epithelium. Here, the average distribution of these angles again skewed towards angles smaller than 90 degrees. And importantly, when sonic hedgehog is inhibited, such orientational bias was absent. This again suggests the critical role of sonic hedgehog signaling in orienting the cells and inducing the directional cell migration. In summary, by developing an ex vivo system to observe and perturb tracheoesophageal separation, we identified an essential and yet underappreciated mesenchymal force attributed to directional cell migration toward the epithelium. We further uncovered the critical role of sonic hedgehog in inducing the directional flow of cells. Our manuscript is recently posted on bioRxiv. In the near future, in collaboration with the Madhavan lab, we are developing a theoretical model to understand the robustness of TES morphogenesis. In particular, we find that as the initial tube constriction happens, it generates a negative curvature which could locally constrain sonic hedgehog diffusion and enrich its concentration, and that could potentially feed back to sonic hedgehog gradient and augment the mesenchymal force. Such positive feedback may explain how or why TES is robust in normal tissue morphogenesis.
1 minute.
Another direction I'm interested in is the epithelial dynamics at the septum, where we know that there is a rapid loss and reestablishment of apical basal polarity for these cells. And genes involved in endocytosis are recently implicated in patients as well as animal models. And I think our ex vivo culture and imaging system would provide a great tool to observe these detailed cellular dynamics to reveal what's really going on at the septum. I would like to thank my advisor, Cliff, and my everyone in the Tabin lab, and my collaborators, Maha, Lodewijk, Nandan, and Alessandro, for the discussion and support throughout this project. I'm happy to take any questions. Thank you.
All right, thank you so much, Ray, for the wonderful talk. So everyone, just remember to put your questions into the Q&A. And while everyone is typing, I have a question for you, if you will. So during your RNA sequencing, when you inhibit hedgehog signaling, you get rid of a mesenchymal population, but what happens to the other population of cells?
Yeah, let me go back to that slide. So we see the composition of other cell types were not dramatically affected, as you can see from this bar plot. Although we could indeed identify differential gene expressions in these cell populations, essentially in all cell populations. But in particular, this mostly affected cell type, the dorsal subepithelial mesenchyme, shows gene regulation, gene expression differences, especially related to extracellular matrix and cell migration, which is consistent with the phenotype we observed.
Interesting, thank you. Okay, we have a question from Irene. Very nice work, elegant experiments. Did you also look to see if there were differences in the biomechanics or composition of the ECM in the two regions, the esophagus and the trachea primordia?
Yeah, that's a really interesting question. And we indeed looked at a few extracellular matrix components in the mouse and tissue samples. Let me find out if I have any example. Like here, we are looking at the hyaluronic acid labeled by the hyaluronic acid binding protein. And in the cheek, it has an enrichment dorsally, while in the mouse, it's more or less uniform. And also, the fibronectin shows very distinct features between the dorsal and ventral foreguards. So these could suggest the mesenchyme itself could have different mechanical properties dorsally and ventrally. And it's actually pretty consistent with our observation that the dorsal mesenchyme is more fluidic than the ventral mesenchyme by having a more disorganized fibronectin as well as more hyaluronic acid.
All right, great. We have another question from Celeste Berg. Does TES share any mechanistic or molecular overlaps with heart development? If not, are there any other organ development processes that could follow similar roles as what you have found? That is, how general is TES?
Yeah, that's a great question. I'm assuming the heart development question you mentioned might be related to the atrial or septation, where you develop a septum to separate the heart into different chambers. I think that's a mechanism a little bit different from TES because that involves a growth of the endothelial cells itself instead of it being squeezed by an external tissue. But this similar epithelial splitting events also happens in the other end of the digestive tract, which is the separation between the anus and the urinary tract in the embryo. And very similar mutations in sonic hedgehog could indeed cause both phenotypes in the anterior foregut and also the posterior hindgut. So
Great.
there are a few cases of such epithelial splitting in vivo.
All right. And another question from Sally Moody. Do patients with TES defects also share other well-described sonic hedgehog defects, such as extra digits or holoproencephaly?
That's a great question. To my knowledge, actually, the TES patients, the TES can be a symptom of many different syndromes, but not in particular with extra digits or holoproencephaly. I guess those are directly related to sonic hedgehog indeed, but patients with those mutations causing these phenotypes may have just a mild functional decrease in sonic hedgehog signaling, not as dramatic as to fully abolish TES. We suppose or we hypothesize that when the sonic hedgehog defect is too strong, it will cause TES as well as many other phenotypes that the infant may not even be able to survive. And this pleiotropy of sonic hedgehog may be in effect. And I think those patients where we could observe extra digits or holoproencephaly might have a milder sonic hedgehog reduction.
All right, thank you. And I actually have another question. So you show that if you perturb migration, then TES is also perturbed. I'm wondering too if there's any defects in perturbation of proliferation of cells as well. Have you looked to see if proliferation also promotes TES?
Yeah, sonic hedgehog definitely has two roles. First, it induces directional cell migration, and second, it promotes the proliferation of dorsal mesenchymal cells, and both can help or facilitate tracheoesophageal separation. But in particular, we tested whether cell proliferation is necessary for TES by doing this experiment. So first, we quantify cell proliferation profile in the tissue, and we found there's no obvious enrichment for cell proliferation in the middle. And again, I think more importantly was that this live imaging experiment with acetylcholine, which is an inhibitor of cell division. And in this case, even when cell division is blocked, we could still see the completion of TES. So that suggests, although cell proliferation may be helpful, it's not required for this to happen.
That's really interesting. We have another question. Is it odd that cyclopamine affects ventral epithelium more than dorsal epithelium, given that it affects dorsal mesenchyme? Thoughts?
I guess we have to go back to the like the drug treatment. I'm not sure what kind of ventral epithelium phenotype you are talking about, but actually in if we do immunostaining of this tissue, it actually shows a dorsal-ventral patterning of the epithelium, and it's just the entire epithelium is a lot smaller than the normal epithelium. So sure, there is reciprocal signaling, like the reduced mesenchyme or perturbed mesenchymal cells can indeed affect the epithelium cells also. But we didn't see a dramatic perturbation in the dorsal-ventral patterning of the epithelium.
Okay. All right. And with that, we have no more questions. So everyone, let's thank Ray one more time. And we will pass it on to Gerrald.
Yes, thanks, Ray, for your excellent talk. And yeah, I will introduce our second speaker of today and moderate the second part of the seminar. So it's my pleasure to introduce Dr. Talia Medwig-Kinney. Talia did her undergraduate studies at Stony Brook University, where she got a degree in biology and health science with a focus on developmental genetics, public health, and community health education. She then proceeded to get her PhD in genetics at Stony Brook University as well, and is now doing her postdoctoral work at the University of North Carolina, Chapel Hill, in the lab of Professor Bob Goldstein. Through her career so far, Talia has obtained several prestigious awards, such as the F31 Predoctoral Fellowship from the NIH and a Postdoctoral Fellowship from the American Cancer Society, as well as many others. Beyond Talia's excellent research and publication record, I'm also impressed by her generous involvement in teaching, service, scientific advocacy, and outreach. In particular, related to Talia's degree in community health education, she has been involved in many outreach events, such as Shadow Scientist, STEAM Night, Winspire, and she's actually the founder of Project WORMS, which stands for Women for Outreach and Role Models in Science. And I think these contributions are especially relevant in current times to exchange the importance of science and research with the public. Unfortunately, Talia couldn't make it live today due to family emergency, but we will show a pre-recording of her talk. And Talia's contact information will be shown in order to ask your questions directly to her after the talk. And with that, I'll give it to Talia's pre-recording and hear about her research.
Hi, everyone. My name is Talia. Thank you so much for the introduction, and thank you to the organizers of the Ethel Browne Harvey Seminar Series. I'm really honored to be among this year's cohort of amazing speakers, and I'm also grateful for the opportunity to be able to share my research with you all today. If you're watching this recording, unfortunately, that means I was not able to attend the live session. However, I've included my contact information at the bottom of this slide, and will show it again at the end of my talk. So please feel free to contact me with any questions or feedback you may have on what I present today. I'm going to be sharing my research on cell shape change, specifically how developmental patterning inputs help to coordinate localization of myosin activating machinery. But before getting into that, I want to take a step back and introduce the broader question that really motivates me as a biologist, and that is, how does cellular diversity arise? If we look within our own bodies, we have a variety of cell types that exhibit tremendous diversity in terms of both form and function, despite these cells having identical genetic blueprints. I'm really interested in how cells use a combination of transcriptional regulation as well as control of protein localization in order to achieve these very distinct cell shapes and behaviors. As a postdoctoral researcher in the Goldstein lab, I study a cell shape change known as apical constriction. Apical constriction refers to shrinkage of the cell apex. This results not only in dramatic changes in the shape of individual cells, but also has broader implications for the architecture of the tissues that these cells are a part of, and it plays an important role in morphogenesis. The forces that drive apical constriction arise from the apical cortex, specifically contraction of actomyosin networks at the apical cortex. Understanding how apical constriction is regulated is important not only just for the sake of understanding biology, but also has implications for human health. So development of our neural tube requires apical constriction, and if this process fails, it can lead to a variety of birth defects, including spina bifida and anencephaly. Additionally, cell shape changes that are similar to apical constriction are also an important part of cell extrusion. Cell extrusion is an important part of epithelial homeostasis, in which epithelial layers can remove cells that are damaged or unhealthy. And if this process fails, it can lead to the formation of cancer. So understanding how apical constriction is regulated can help us not only understand normal development, but also get invaluable insights into these disease states. So in the lab, we use the model organism C. elegans to study apical constriction. C. elegans has a rapid generation time, it's amenable to genetic perturbations, and it also is transparent, which allows us to visualize cell shape changes in real time. I study C. elegans at the early embryonic stage, and specifically focus on these two cells. These are the endoderm precursors that are going to give rise to the entire gut of the adult animal. Just like our gut needs to be on the inside of our body, the same is true with these worms, but these cells are initially born on the outside of the embryo. As you can probably imagine based on the theme of this talk so far, these cells move their way into the inside of the embryo through the cell shape change of apical constriction. And these provide a really great model of apical constriction given its simplicity of being only two cells that are large, so we can visualize what's happening within them. So the first question I was interested in when I joined the lab is, how are actomyosin networks organized during apical constriction? When you consult the literature, there's different models of apical constriction, and I'm going to show them below. First, we're going to take a slice through the apical cortex and look from a ventral view. So one model suggests that actomyosin networks are enriched at cell-cell junctions, and this generates a force that resembles that of cinching a purse string closed. Another model that was observed in ventral furrow cells of Drosophila by Adam Martin's lab suggests that myosin and myosin activating kinases are enriched at the center of the cell apex, and that actin filaments are radially polarized, representing or resembling the organization of actin, myosin, and sarcomeres. Another possibility is that actin and myosin are diffusely localized throughout the apical cortex, which would generate a more uniform force applied throughout. So to address this, I collaborated with a very talented graduate student in the lab, Rui Yan, and we took advantage of C. elegans strengths for live cell imaging and looked at endogenously tagged components of the actomyosin network. So in this movie, we're looking at a time lapse of apical constriction where the two endoderm precursor cells are briefly highlighted in blue. The magenta being shown is representing a plasma membrane marker, so you can look at the location of cells and their shapes. And myosin, or non-muscle myosin 2, NMY2, is being shown here in green. So in this figure where I'm showing screenshots of the movie, the top row is showing that plasma membrane marker where you can visualize the location of cells. The endoderm precursors are marked with yellow arrowheads. And the second row is what I want to draw your attention to. So this is where we're looking at localization of non-muscle myosin 2. And the third row is showing an overlay between the two channels. So it appears that non-muscle myosin 2 does not have any central enrichment nor enrichment at the cell-cell junctions. It appears to be pretty broadly localized throughout the apex, but we wanted to be quantitative in our observations. And so what we did is we took these movies, we collected several measurements spanning the cell apex from different directions, and then we, using multiple samples, we pulled this data together to see if there were any broader patterns. Now, in the case of myosin, because of its punctate nature, this data is a little bit noisy, but hopefully you can appreciate that there's no consensus pattern of enrichment of myosin in the apical cortex. That doesn't mean that myosin activity is broadly distributed, though. So to test for myosin activity, we looked at this myosin activating kinase, MRCK1. So again, showing you the screenshots of this movie, we can see that MRCK1 is broadly distributed throughout the apical cortex of the endoderm precursors, just as we saw with myosin. And this is reflected in the quantitative data shown below as well. Next, we turned our attention to actin filaments, and we wanted to visualize the polarity of them. To do so, we utilized these endogenously tagged capping proteins, the barbed end capping protein CAP1 and the pointed end capping protein UNC94. When we looked at CAP1, we saw that there was enrichment at cell-cell contacts, which is similar to what was observed in the Drosophila ventral furrow. However, we did not see central enrichment of the pointed end capping protein UNC94, and instead it appeared to be pretty uniformly distributed throughout the apex of these cells. This is suggestive of more of a mixed polarity network of actin. So again, comparing to what was observed in Drosophila, where myosin and its activator are centrally enriched in the apex and actin filaments are radially polarized, we instead observed a more diffusely organized cortical actin-myosin network driving apical constriction in C. elegans. And based on our review of the literature, we think that this may be more reflective of what is observed in other vertebrate models. And so, taken together, this data suggests that there are multiple strategies for achieving the same morphogenetic outcomes. So after we defined the architecture of the actin-myosin network driving apical constriction, I next turned my attention to asking, how is cell shape change coordinated in space and time? So put more simply, how do these cells know to change their shape, whereas their neighbors don't? And I think that the answer lies in regulation of MRCK1, the myosin activating kinase. I showed you films of MRCK1 localization during apical constriction a few slides ago, but I think it's more striking to look from a lateral view. So in this case, the two endoderm precursor cells, again marked in yellow arrowheads, instead of internalizing into the screen, they're actually going to be moving upwards. And in this view, I think it's very obvious that you can see apical enrichment of MRCK1 in these two cells, but not anywhere else, despite MRCK1 being broadly expressed throughout the embryo. So this led to the mystery, how? How is MRCK1 localized and apically enriched in these cells in particular? We know that MRCK1 interacts with CDC42 via its CDC42 and RAC interactive binding domain, or CRIB domain. CDC42 is thought to be inactivated at sites of cell contact, leaving its active form at the cell apex. I wanted to know if CDC42's interaction with MRCK1 could explain its apical localization in these cells. So again, I collaborated with Pooh, and together we looked at localization of other CRIB-containing proteins, including ARC1, MEX2, PAC1, and WASP1. And interestingly, we did not observe any apical enrichment of these other CRIB-containing proteins. And in fact, there doesn't seem to be a consensus localization pattern at all. Some of these were enriched at cell contacts, some enriched at specific cell contacts, and others were enriched or localized throughout the cytoplasm. And so this data, combined with the fact that CDC42 is also expressed broadly throughout the embryo, tells me that this cannot fully explain MRCK1's localization. We do know, however, that MRCK1 apical enrichment is downstream of endoderm cell fate based on previous work that was done in the lab. So these endoderm cells are specified by these two mostly redundant transcription factors, END1 and END3. And if you perturb both of these proteins, so in this case, we're looking at a mutant of END1 treated with END3 RNA interference, we see that that apical enrichment of MRCK1 is lost. And so in order to identify candidates that could potentially be regulating MRCK1 localization, we want to look for factors that are downstream of endoderm cell fate that could be affecting MRCK1 localization. One hypothesis is that these transcription factors are turning on genes that promote MRCK1 localization and/or its interaction with CDC42. Alternatively, there could be factors that are normally inhibiting MRCK1 apical enrichment that are being turned off specifically in these cells. So in order to identify candidates, we're taking a multi-pronged approach where we're using transcriptomic data to identify transcripts that are downstream of endoderm-specifying transcription factors. We're also looking at transcripts that are specifically up or down regulated in the endoderm precursor cells. And we're looking at proteins that are predicted to interact with MRCK1 and/or CDC42. And we think that the overlap of these data sets will give us a candidate list that we could then screen through RNA interference to see if their knockdown disrupts MRCK1 localization. So for this part of the project, I'm collaborating with a very talented undergraduate student named Jaden Perez, and we are working with unpublished RNA sequencing data from Sophie Tintori and Jessica Sullivan Brown, former members of the Goldstein Lab. So I'm going to walk you through this data now. Each of these blue points represents a single transcript that is expressed in the C. elegans early embryo. And the ones that I've highlighted in magenta are the 1,179 transcripts that we've identified as being positively regulated by END3. This is based on RNA sequencing data comparing wild-type animals to END3 mutants. We further narrowed this list down by looking at those that are specifically enriched in E, the cell that gives rise to the two endoderm precursors. This is based on published single-cell RNA sequencing data from the lab. And this allowed us to focus in on 155 transcripts. That's still a long list, though, for a screen that's not very high throughput. So we decided to prioritize this list using protein-protein interaction predictions using AlphaFold 3. So we took those 155 transcripts. Of those that encode proteins, we took their amino acid sequences and put them into AlphaFold 3 and tested their ability to interact with the membrane binding domain of MRCK1, just shown here, and also separately tested their ability to interact with CDC42. We could then take the confidence measures of these predicted interactions and use them to prioritize our list of candidates. So I'll come back to that screen in a bit, but I also want to talk about some efforts that we're working on in parallel. So I'm working with another talented undergraduate student, April Song, who's actually graduating today, and we wanted to identify the MRCK1 domains that are important for localization. So within this membrane binding domain, there are several smaller domains, including a C1 domain, a citrin homology domain, a plextrin homology domain, and a crib motif, which is part of a larger P21 binding domain. And in order to test which of these are sufficient for apical localization, we decided to break up this membrane binding domain and attach each of these subdomains to a fluorescent protein to visualize its localization. And so these are the results of that experiment. In the first row, I'm again showing you a plasma membrane marker so you can look at the cells and their positions. I'm highlighting the endoderm precursor cells with those yellow arrowheads. In the middle row, which is what I want to draw your attention to, is the localization of these transgenes. Please note that these were all inserted into the genome as single copy. They're all driven under the same MRCK1 promoter, and they were all captured using identical imaging parameters. So any changes in intensity between them is likely due to differences in protein stability. And so for the dim images, we included enhanced versions in the top right corner so you can more easily visualize their localization. So each of these domains has slightly different localization patterns. Sometimes we see expression in the nucleus, other times it's just in the cell-cell contacts, other times it's both. But what we did see, or I should say what we didn't see, was any apical enrichment. And so we weren't completely surprised by this result because we hypothesized that perhaps multiple domains in combination are required for apical enrichment of MRCK1. But what was a surprising result is when we looked at our control strain, which was the membrane binding domain altogether fused to a fluorescent protein, it also did not exhibit apical enrichment. And so this This is surprising because we assumed that all of the information that governs the localization of the full-length protein exists in the membrane binding domain. And so the fact that this transgene did not recapitulate localization of the full-length protein suggests one of two things to us. Either the kinase activity of this protein is important for its localization, which would be unexpected, or alternatively, the dimerization of this protein is important for its localization, which we were not able to recapitulate by just looking at the membrane binding domain alone. And so this leads us to some future directions that we're currently working towards. First, we're generating a full-length kinase-dead MRCK1 transgene. This will help us differentiate between whether the kinase domain is important for its localization or whether dimerization of the protein is important. We're also planning to repeat the AlphaFold 3 screen with 2 copies of the full-length MRCK1 protein to make sure that we're not missing out on any important any important interactions. And finally, the second half of the story, we've been focusing on myosin regulation. However, the lab is also interested in how actin regulation contributes to apical constriction. And so there are multiple projects in the lab led by Pooh, and we have collaborative efforts, including characterizing different actin probes, which we hope will be a useful resource for the community. And so with that, I would like to thank my lab, especially my supportive mentor, Bob, as well as Pooh, April, Jaden, and former lab members, Ella, Jessica Sullivan-Brown, and Sophie Tintori for their contributions to this work. I want to thank my funding and fellowships. And I also want to, again, include my contact information here. Please send me any questions or feedback you may have, and I'd be happy to reply. Thank you so much for your attention, and have a great day.
All right. Thanks, Peter, for your talk, and for you not live here to get our questions and appreciation for your talk. But again, contact her directly through her email or social media that she presented on the beginning of her slides. And I want to thank everyone who is here. I want to, of course, thank Dr. Rui Yan, Dr. Talia Medwig-Kinney for their excellent talks today. Again, both seminars have been recorded, will be available on the SDB website next week. And of course, we want to thank all of the Ethel Browne Harvey Postdoctoral seminar speakers of this season. This is the last seminar of this season, so you have to wait until next year or next season to see the new talks. And applications for the new season, the 2025-26 season, will open in June, so keep an eye on that and let your postdocs know, let them sign up. Thank you all for coming, and have a wonderful day.